Chronic pain is not just a medical condition, it's a life-altering experience that affects millions globally, ranking as one of the top causes of disability. This persistent pain can complicate everyday activities, placing immense personal and economic strain on individuals and society as a whole. While researchers have long proposed theories regarding the underlying molecular mechanisms of chronic pain, pinpointing the exact biological processes involved has remained elusive until now. In a groundbreaking study, a collaborative team led by Professor David Bennett from the Nuffield Department of Clinical Neurosciences, NDCN, and Professor Simon Newstead from the Department of Biochemistry and the Kavli Institute for Nanoscience Discovery has made significant strides. They have uncovered a new genetic connection to pain, elucidated the structure of the molecular transporter encoded by this gene, and established its role in pain perception. This research, generously funded by Wellcome and supported by the NIHR Oxford Health Biomedical Research Centre, OHBRC, presents a promising new target for drug development aimed at alleviating chronic pain. In many cases of chronic pain, nociceptors, specialized nerve cells that sense tissue damage, become overly active, transmitting excessive pain signals to the brain. This heightened signaling can lead to an experience of pain that is more intense than what would typically be expected. Although the precise regulation of these signals is still not fully understood, some studies have suggested a link between these changes and polyamines, natural compounds produced by the body that assist in various cellular functions. Over time, an accumulation of polyamines may lead to the oversensitization of nerve cells, resulting in long-lasting damage and chronic pain, where even minor stimuli can provoke significant discomfort. However, these theories have remained largely unverified until now. The absence of a clearly defined target has made treating chronic pain particularly challenging, often resulting in a reliance on powerful opioids. While these medications can effectively reduce pain, they interact with multiple pathways in the brain and carry a risk of addiction, leading to serious long-term health consequences. Professor David Bennett emphasized the urgency of this issue, stating, "Chronic pain is an escalating societal challenge, and current treatment options are inadequate. We must deepen our understanding of the mechanisms that drive chronic pain in humans and, crucially, identify new targets for analgesic drugs." To explore why some individuals experience chronic pain more acutely than others, the research team utilized data from the UK Biobank, comparing genetic information with responses from participants regarding their pain experiences. They discovered that individuals with a variant of the SLC45A4 gene were more likely to report heightened pain levels. This finding was corroborated by data from other significant population studies, including FinGen. The next step for the researchers was to investigate the function of the SLC45A4 gene. Dr. Stephen Middleton, the lead author of the study and a senior postdoctoral researcher, shared his excitement. Establishing a connection between SLC45A4 and chronic pain in humans was thrilling, but the real challenge was to decipher the gene's role in the body. We were astonished to find that SLC45A4 encodes the long-sought neuronal polyamine transporter, which plays a crucial role in how certain nerves react to painful stimuli. This discovery has expanded our understanding of pain signaling and opened new research pathways for chronic pain treatment. Our work showcases the power of collaborative scientific discovery. Working alongside Professor Simon Newstead and employing cryo-electron microscopy, the team successfully mapped the 3D structure of the transporter in humans for the first time, confirming its role in transporting polyamines across nerve cells. Additionally, the researchers found that this gene is highly expressed in the dorsal root ganglion, a critical area where sensory neurons relay information from the skin and muscles. The nerve cells in this region are pivotal in detecting pain, and the volume of signals sent to the brain is essential for modulating our pain responses. In experiments with mice that lacked the SLC45A4 gene, a gene they share with humans, the animals exhibited a diminished response to standard pain stimuli. While the mouse nervous system is not an exact replica of the human system, many fundamental mechanisms are conserved across species, suggesting promising avenues for future research on the SLC45A4 gene. Professor Newstead remarked, "Major breakthroughs occur when we comprehend how the intricate tissues and organs in our bodies function and interact. Membrane transporters are vital to this communication. Our findings unveil a new connection between membrane transport and chronic pain, paving the way for a deeper understanding of how metabolism and pain are intertwined in the human body. With continued research, the potential exists to develop a successful drug that could alleviate chronic pain without the need for strong opioids, leading to safer and more effective treatment options for patients around the world." In conclusion, Professor Bennett stated, "We have identified a new gene associated with pain, gained insights into the atomic structure of this molecule, and linked its function to the excitability of neurons that respond to tissue injury. Ultimately, our discoveries reveal a promising new target for chronic pain treatment." The complete study, titled SLC45A4, is a pain gene encoding a neuronal polyamine transporter that's been published in the journal Nature.